Hi everyone, today I'm going to be presenting the paper for this week, which is brain region specific organoids using mini-bioreactors for modeling Zika virus exposure. For the following presentation, I'll be following the next outline. I'm going to start with a brief introduction to organoids, brain development, and Zika virus. Then I'm going to present you the hypothesis of the paper. We'll be following the experimental design, the results of that experimental design, the limitations of the experiments, and some possible future directions for the paper or for the study. Okay, I'm gonna start with the Zika virus. It was discovered in 1947 in Africa and Asia. It was believed to be harmless and it is transmitted by a mosquito. This is a type of flybovirus, which is like dengue and yellow fever. So the symptoms in adults, they are usually flu-like symptoms, but most of the people that are transmitted with Zika virus do not present any symptom. In the map in pink, we can see the regions of the world in which Zika virus is currently active. We can see that now it is spread to Central and South America. So this leads us to the timeline of Zika virus. So as I mentioned, it was discovered in 1947 in Africa. So the Zika virus was first detected for the first time in Brazil in May of 2015. I want to mention that in 2014, in June, the World Cup was held in Brazil. So it is believed that travelers from Africa and Asia carry the virus to America. At the end of that year, in November, an increment in microcephaly cases was reported in Brazil, which near to 4,000 new cases. Just to set this, in 2014, the reported cases was around 150. So by January of 2016, the World Health Organization declared an international public health emergency because Zika virus was expanding over Central and South America. So it was believed that the increase in cases of microcephaly was due to Zika virus, but no correlation was confirmed to that date. So by March of 2016, the Zika virus 3D structure was discovered and was published. And by May of 2016, the infection mechanism of Zika virus was studied by several groups around the world. Some characteristics of the people that suffer microcephaly are delayed development, failure to thrive, learning disability, short stature, or slow growth. Also hyperactivity, impulsivity, and irritability, as well as malnutrition, poor coordination, and other types of impairments. As a refresher for this week's lecture, I'm gonna go over gastrulation process really quickly. So we have our embryo here, and we know that we have our epiblast, and that the cells of the epiblast will start to differentiate and migrate, and eventually the neural tube is gonna form. Here we have the frontal view and the side view. So this is the neural tube. We know that the neural tube, it's gonna later be divided in forebrain, which is more or less like this, midbrain, hindbrain, and the spinal cord. Now let's go a little bit deeper in the development of the forebrain, which is this part of the neural tube, the part in yellow here. If we take a slice of this tissue, we are gonna find primarily neural embryonic stem cells. So these cells are going to start to proliferate and differentiate to give place to which is gonna later gonna be the cerebrum of the fetus or the person. As the embryo starts to develop more complexity in this tissue in the forebrain, it's gonna be gained. So we're gonna see how the stem cells from here are gonna start to form different tissue or different types of neuronal cells. By the week 13 after implantation, we're gonna start to find separation or layers in the tissue. So if we again do a slice of the tissue, we're gonna be able to see all these populations of different cells, which of course they have different cell markers. Then around the 26 weeks after implantation, we're gonna find this very complex tissue. This is gonna be the innermost part of the brain, which is gonna be somewhere here, and it moves out. So we can see that we have different types of cells, of which I'm gonna talk in more detail in a few minutes. So going back to Zika virus, we know that we have this very complex process of differentiation and proliferation, and that by the end of the week 30, we're going to have this very complex tissue formed by several types of neural cells. So what we don't know is in which stage Zika virus infects the cells, and we also don't know what type of cells the Zika virus is more likely to attack. When this article was published, there was a lack of physiologically relevant models that can be used to model infection of developing brain. These studies were published around the same time of this paper. The first one used monolayer cell culture or 2D culture. They used Zika virus and tried to assess which types of cells the virus is more likely to attack. There also was another study that developed neural spheroids. At the end of both studies, the authors conclude that there is an increase in cell death and there is a decrease in size of the spheroids. So there is a strong evidence that Zika virus can actually cause microcephaly by this time. However, the lack of complexity of these tissues didn't help to elucidate the mechanism in which the Zika virus is involved. So to recreate physiologically relevant tissues, well, we know that on the regular development process of the brain, we have activation of several pathways such as Wnt and SHH that will help with the patterning of the brain. In vitro is a little bit more complicated to reproduce this. There are several methods. One of the most used is the neural induction of pluripotent stem cells, either embryonic or induced pluripotent stem cells. These cells can be either floating or can be suspended in matrigel. For this case, the authors used the floating methods, to which they develop a new bioreactor, and this will lead to self-organization of the cells, hopefully. So with this, I conclude the introduction. Just to recapitulate, there is a strong evidence that leads to suspect that Zika virus can cause microcephaly. We review a little bit of brain development, specifically the cortical development of the brain, and we review how some in vitro tissues can be made to replicate brain models.